Thank you very much, Kathrin, for this nice introduction. And I'm now going to tell you about an animal-free cell culture, because we have to replace fetal bovine serum to receive a humane and robust science. As Kathrin said, we published in August a big review article about leaving behind fetal bovine serum, and much of it I will talk about now in this talk. But I will start, for everyone who is not familiar with fetal bovine serum and the issues with it, I will start with a big background. So we start with a simple experiment. Two labs are performing the exact same experiment. They are using A549 cells and cultivate them under the same conditions. They use the cell culture medium and the supplements from the same suppliers, and they were of course expecting that the results would be similar. However, in the end, the results were highly variable. So what has happened? Apparently, the experiment was not so simple. So they did what all good scientists do. They repeated the experiment again. And this time they were using the same lot of the cell culture supplement. So they wrote here in their paper. And now they had a low variability of the results. So what do we learn from this? Different lots from the same product and the same supplier can still differ so much that it influences the experimental results. And this, of course, is part of the big reproducibility crisis we're all facing. So why do replicates fail? Why do we have this reproducibility crisis? Well, of course, we have the infamous publish or perish mentality, which means that you are rewarded if you publish a lot and fast, and not so much if you also publish good. And also, science tends to publish mostly positive results. Negative results, not so many people want to see, especially not when they actually have no time. And big issue is non-standardized methods that are used and also non-standardized materials. So, but what do I mean with that? Well, non-standardized materials, of course, animal experiments. As we all know, animal experiments are black boxes. We are not sure what's really happening in there. And we have a big issue with the transferability because we want to use rats to find out medicinal research for humans. And also these animals live in a highly artificial environment, and there's a lot of variability. They have variability between the labs, inside of the lab, between the technicians who are making the experiments with the animals, and also, of course, the different animals are still individuals, even if they are identical twins. So, but also in vitro experiments. So cell culture has also problems because many of them use still non-standardized materials. Even though you have the advantage that in cell culture you can use the cells and cultivate them outside of the body, but actually from the subject that you actually want to study. Like if you want to research a medicine for humans, you can use human cells. So you have the target species, but they still live in an artificial environment. And there's, of course, also variability between the labs, as we've shown before. So an issue there is that they use mostly animal-derived materials, which, as said before, are irreproducible many times. So these cells would still be inside of a black box. So what are we going to do now? We have to light up the black box. Because as long as non-standardized materials are used, the cells would also stay in the black box. But luckily, it is possible to replace these materials with materials that have a disclosed composition. Because only then, when we light up this black box, we can focus on studying the really important question. What is happening inside of the cell? Unfortunately, the use of animal-derived materials is still routine in many cell culture labs, even in methods that are aimed to replace the use of live animals. Here you can see a technical report from the Joint Research Center of the European Commission. They looked into thyroid in vitro methods, and they found that only in the cell-free assays there were no animal-derived ingredients. However, in all of the other cell culture methods, at least one animal-derived material was included, and all of them had fetal bovine serum in them. So this is clearly the biggest issue. Therefore, I'm going to focus on this topic here mainly. So for people who don't know, what is fetal bovine serum? But sometimes you also know it as fetal calf serum, so FBS or FCS, but I'm going to stay here with FBS. FBS is a widely used cell culture media supplement. It's really loose since the 1950s, and it's hard to find any cell culture that not at least was cultured once with FBS. Because this FBS is so rich of nutrients and growth hormones, which is helpful for the proliferation of cells. But as you can see here on the side, that FBS doesn't come out of nothing. It's a byproduct of the beef and dairy industry. It in fact comes from the blood of fetuses that are derived from slaughtered pregnant cows. A needle is introduced into the heart of the fetuses after the slaughter of the dam, and the blood is received. And it is not mandatory to kill the animals before that or to apply any anesthesia. Sometimes the animals are just being left there to die of asphyxiation before the procedure starts. Sometimes they're still alive, but this is removed. I'm not going to show here any videos or pictures of that, but I'm going to link here a video from undercover research in France from 2021, where these procedures are documented, despite other features. But viewers with discretion is advised. So now I talked a lot about FBS, but what would actually happen if I would use FBS in my cell culture? Well, the FBS could alter the model system that I'm using, because FBS, as the name says, it comes from a fetus. Therefore, it contains a lot of hormones and growth factors that are related to the development of fetuses. And most of the time, you have adult cells or other cells that are not fetal bovine cells. Therefore, you would definitely have an effect on these cells and might even alter them, especially when you're using stem cells, because the FBS can induce unwanted reprogramming and also unwanted differentiation of these stem cells. For example, there was a study where they tried to engineer skeletal muscles from murine cells, and the FBS that they used in the medium altered the whole muscle phenotype. So this is something you do not really want in your cell culture, especially not in your stem cells. Another issue is that FBS can be contaminated with pathogens, especially viruses, but also prions, bacteria, fungi, endotoxins, and even extracellular vesicles have been found in FBS. And this is, of course, something that you put in your cells, and maybe in the end, you put it into the patients if you would use these cells. Studies shown that even low levels of serum can promote a rapid growth of mycoplasma in your sample. And even if nothing happens, if no pathogens are in your sample, there are still some genogenic substances because because the FBS is from a non-human animal. And there's also studies that show that human cells can incorporate xenogenic substances from the FBS, which can have, of course, in the end, problems when you have a stem cell transplantation. And they have like substance in it that could cause an immunogenic reaction. Unfortunately, the production of FBS is not transparent. The production steps are challenging to retrace, despite many traceability efforts that have been applied. So I can show you here a ratio of where the FBS is coming, but this is still very broad and only an estimation. You can see that when this was done, approximately around 2020, the USA is at 26% of the worldwide production of FBS, followed by Brazil, and then the other countries of the Americas, and followed by Australia, Europe, New Zealand, Africa, and other countries. You can see most of them comes actually from the American continents. There are, of course, also only estimations of the global animal FBS production available. And according to our research, it's estimated to be 800,000 liters per year, which corresponds to 1 to 2 million calves. We cannot really say how many calves it is because depending on the age on the time of gestation of the fetuses, you could retrieve more blood from a fetus. So therefore, we would say 1 to 2 million calves each year. And as you know, if something is not transparent, it could give incentives for fraud. And there were scandals in the past, or maybe even now that we not know of yet, where they were putting other materials into the serum to sell it for a better price, or even to mislabel the origin. As you can see on the table on the side, New Zealand is about 5% of the global production, but there was 1 year where they found more than double of New Zealand labeled FBS on the market than New Zealand is actually producing. Because New Zealand is known for being, at least on paper, FBS free and BSE free. Therefore, it was decided to have this. You can sell it for bigger prices. Therefore, there was a lot of incentive to actually mislabel it. So this, of course, is having troubles for your research if you cannot be sure what you're putting inside of your sample. And as you can see, it's still from a lot of different countries and from different regions inside of these countries. Therefore, this will even increase the reproducibility issues of FBS lots even more. So FBS can also be uneconomical if you're using it, because the prices are inherently volatile, because they are interconnected with the price and the supply of beef and dairy products. And there are many issues that could affect the price of agricultural products, like animal pathogens, natural disasters, changing policies on farming, and right now in our time and age, trade sanctions and tariffs. All of this will affect the prices of FBS. And as you can see, costs are often the key, and the uncertainty will dampen your long-term cost calculations of your project when you cannot be sure if you maybe have to pay double as much for the FBS that you want to buy and you don't have the money. This might even slow down or terminate the whole project if there's a certain price surge. So, and finally, FBS, the use of FBS results in an ethical dilemma, because FBS is produced from fetal blood, which has always been taken invasively at slaughterhouses. And there's no scientific consensus yet on whether the bovine fetuses would suffer during or after the slaughter of the pregnant cow. However, we should give the animals the benefit of the doubt, just as we give humans the benefit of the doubt, because also there is no 100% consensus if human fetuses can suffer while they're in the womb. But just for that, we still give anesthesia to the mother and the baby when there is any in utero surgeries. And we should give this benefit of the doubt also to cows, especially since cows are highly developed when they're born. When they are born, they can walk and run away on the same day, which is something that humans cannot do for a long time. And of course, not only the calves suffer, also pregnant animals suffer a lot when they were transported, especially when they are in the end of their gestation. Therefore, they should not be transported to slaughterhouses at all. And in this day and age, it's also possible to test if cows are pregnant or not. So they should not be transported at all there. So now I still want to use FBS, but what can I do? What are the common countermeasures that people tell me when I still want to continue to use FBS? Well, one common countermeasure is to just ignore the issues. But as we all know, this hardly solves any problems, and all of them will come back later to haunt your project. Another countermeasure is that you do an extensive pre-testing of the FBS batches, meaning you know you have a certain big project, you want to use like say 10 liters of FBS. You're not sure if you should really buy this batch, so you buy a lot of small batches, test them, and the best one you will then save and buy all the 10 liters, so even more of this. And then you do all your results or all your experiments with this batch. This is, of course, quite expensive. And what happens if you run out of the batch, or if it's not anymore available, or there are only 5 liters instead of 10? And also, are you still writing the lots of the FBS that you're using in your papers or not? So another countermeasure is that people suggest that we use more laborious manufacturing techniques, like more filters, more pooling, more antibody removal to create a pure or a cleaner version of FBS. But just like with the pre-testing of the FBS, this will all result just in higher costs for the end consumer. And FBS is still an inherently undefined product. So whatever you do, you will only define it to a certain extent. Therefore, it will always be a lose-lose situation if you use FBS, but not only for the science, also for the animals and the ethics. But luckily, I can tell you that the FBS can be replaced. There are numerous replacement materials available. For example, you can look at the FCS-free database, which is hosted by the 3R Center of the Utrecht University in the Netherlands, where you can see so far, I think, over 800 different entries in this database. You will find a lot of commercially available products on the market. And there are open access formulations for cell culture media that can be found in the scientific literature. However, the second and third point have some caveats, which I will come back later to. So now, after the background and the sum up of the problems of FBS, now I'm going to tell you about applications of FBS-free media. All these topics we mentioned in our review. We will start with the stem cells. And as you know, for stem cells, they are very fragile. The undefined components of FBS can cause a spontaneous differentiation, especially when you use human stem cells. And all of this will make it very difficult to maintain the undifferentiated state. And there are some solutions for that. One solution would be human platelet lysate, and the other one would be chemically defined media. Let's start with the human platelet lysate. This is being done by using platelet donations to freeze them and then unfreeze them, and therefore all the platelets are lysated, so they cracked open and they relieve their inners, and this is then centrifuged and used as the human platelet lysate. These are, of course, because they're human-derived, they are ethically acceptable, but only when they are derived from voluntary donors and only if these donated batches are expired. Because platelet donations are only have a shelf life for 3 to 5 days. Since these platelet lysates are used and designated for medical uses in humans, they have a very low pathogen and contamination risk because they were extensively tested before that. But of course, the risk is never zero in this case, like with all blood products. When you use them for human cells, they are xeno-free and almost universally usable. But of course, if you have other cells like rabbit cells, a human medium would still be xenogenic. Especially for mesenchymal stroma cells, HPL is very good to maintain the characteristics of these cells, and so they can be used for isolation, expansion, and cryopreservation. And it also improves the functionality and longevity of the CAR-T cells. However, as we are all like walking, talking blood banks, and we are all different, also the human platelet lysates is still undefined. And it is hard to scale up the production if you don't want to get in competition with the medical use. Therefore, chemically defined media is preferred. Chemically defined media is ethically acceptable, and it is defined, as the name suggests, it's xeno-free and free of pathogens. But, big but is, when the composition is also undisclosed. If you know what's actually inside of this chemically defined medium. Because many companies do not really say what is in it, so you cannot be sure what they're actually using in it. Is it really animal-free? Is it really being tested? And especially, is it the same when I buy it in 5 years? So, when you know what's in it, of course, you could also do it yourself if you have the time and the resources to do it. But also you can modify it yourself because you know like what is in the chemically defined medium, and maybe you need one certain protein that you have to recombinantly produce and put in, and then it works even better. And for human ESCs and iPSCs, the use of chemically defined medium is the only option if you want to ensure the controlled differentiation and also prevent the spontaneous differentiation into unwanted lineages. However, the problem with chemically defined medium is that there's so much out there, and you have to find the right one for your cell. So, the next topic are complex in vitro models. These models are used to study higher complex systems. And therefore, because they themselves are more complex, they can do a closer assessment of real-life physiological conditions. And because they are so complex, all external factors that would influence or threaten the integrity of these systems need to be replaced, best with defined materials. Because the more factors you have, the more complex your system are, the more prone it is for disturbance. And actually, if you want to consider your in vitro complex in vitro models to be fully fit for purpose, you should definitely be independent from animal-derived materials. So what are complex in vitro models actually? Because this is a kind of new term. I can tell you here that we're talking mainly about the 3D microtissues, meaning cells and organoids, but 3D bioprinted tissues and microfluidic systems, also known as organ-on-chips. So I will start with the cells and organoids. There's a study that generated retinal organoids, and they replaced FBS and matrigel with human platelet lysate and recombinant laminin. And also previously to the forming of the organoids, they had to culture the ESCs, and they used xeno-free essential 8 medium that was commercially available. Because this is important, because many times you see that people write in their papers, we use this serum-free medium, but like few lines before you said we just routine cultured the cells before we did the organoids in FBS. And actually, this is mostly the time where you use the most FBS in the routine culturing. So if you do one thing, you should do both. And there's also the Earth's universal replacement medium, short called our medium, that I helped to publish. And I'm also making a little bit of advertisement for this, which is not a commercially available product, but it can be used as an open access medium, where you can also have the formulation and everything inside of it all available here over this QR code. And also a few months ago, there came out a brand new paper that used this medium for the culture of a 3D primary hepatocyte steroids. And we have many reasons to believe that it will also be used for organoids in the future. So now coming to the topic of bioprinting. Here I will deviate a little bit from FBS, because FBS is only part of what you need for bioprinting, because what you mostly need is that you have to have a structure or a hydrogel to keep the printing results in form. And therefore you would use animal-derived materials, but as an animal-derived replacement, you can use cellulose-based hydrogels or alginates from algae, agarose, and even synthetic peptides, like these examples here on the screen. And you can even use extracellular matrices from tissues that are donated, like from other surgeries, and decellularize them and use them as bioinks. And all of these are the cells in there also are many times cultured in a culture medium, of course, and therefore you should also replace them with another medium, because many times the use of FBS is masking the issues that other animal-derived hydrogels are causing to the medium. So you have to do both of them. And there are sometimes also cross advantages, like there was a research that found that you can use proteins from human platelet lysate to make a scaffold, which is also usable for biocompatible materials in bioprinting. Sorry, my throat is a little bit dry. And yeah, the next point of the complex in vitro models are the organ-on-chip models. Many of them use human serum, especially when you have microvessel networks, because this is also what the cells are in contact, or especially the organs are in contact anyway with human serum. However, the problem with human serum is similar to the issues with HPL. So, as I said before, like some reproducibility issues or even the possibility of pathogens. And also you have to replace the heparin that is many times necessary to keep the human serum in flow. Therefore, also the use of chemically defined medium is recommended. So the next point is a big one that is coming out every year into the news. It's the production of cultivated meat. So basically, we have to feed cells to later feed the world with the meat that they are producing. What I really have to disclose, what you probably all know anyway, when I'm talking about xeno-free medium, the use of FBS and bovine cells is actually not a xenogenic problem, because it's from the same species. But of course, as you produce cultivated meat to replace animal suffering, it would be very obvious not suitable if you would use FBS because it still contains animal suffering. So therefore, this industry has to replace the FBS, otherwise it makes no sense to produce it at all. And there's already some medium out there, like the BV9+ medium, which uses as a protein source, they use recombinant human serum albumin. However, I'm a bit surprised that they are not thought about using recombinant bovine serum albumin. This would also make it more species specific and maybe easier for humans to accept it as a food. So another option of the BV9+ is the BVR version, where they just skipped the albumin at all and replaced it by rapeseed protein, which was isolated from agricultural waste, which makes, of course, a lot of sense because if you use rapeseed protein or other plants that you can use later to feed your cells, it would just mirror the same way as if the cows produce their own proteins by eating plants. And there are also different other materials available like metacellulose or racemic alanine, which can enhance the propagation of the muscle cells, because this is what you actually want to eat in the end. So the problem is also that, of course, cultivated meat benefits a lot from the advances of the biomedical industry in the last decades, but there are hopes that also can be the other way around, that the science in biomedical can thrive from the innovations been done by the cultivated meat industry. But there's a lot of discouragement to publish the media formulations because of competitive and proprietary concerns, because companies maybe want to keep their perfect medium for themselves. I can understand that from a financial point, of course, but on the other hand, the transparency on food will definitely build consumer trust in the safety and the nutrition factors of the population, because many people do not want to eat something where they do not know what is in it. And then you can still sell this to companies to produce serum-free medium. So there can be a win-win situation for all of us. So FBS is also used in the production of biotechnological proteins like antibodies. Especially CHO and HEC293 cells are used to produce antibodies because they are matching human glycosylation patterns and the post-translational modification. For the HEC293 cells, it's not surprising because they're human cells. It's a bit surprising that this works for the Chinese hamster ovi cells as well. But still, they are long-time used cell culture that are not like being repeatedly harvested from animals. Therefore, it's more ethically acceptable to use them. These two cell lines are already optimized for bioreactors and in most cases already adapted to a FBS-free medium. Therefore, many commercially available media are already out there on the market, especially for these applications. Also, vaccines are produced in cells, and especially in suspension cells that are best for production in large-scale bioreactors. And there they're also using other cells like HEC923 cells, but also canine bite cells like MTCK, simian-based cells from virus, and even insect cells like SF9 cells. And for all of these cells, an animal-free media is available in the literature. And all of them contain proteins either from soy, yeast, human, or recombinant sources. However, especially for vaccines, a protein-free medium is is recommended because it reduces the extraneous antigens, and also it would avoid an unstable amino acid composition in the medium, because the protein that is in the medium could degrade and could therefore influence the quality of the vaccines. But luckily, also many commercially available media are on the market. I'm not going to list all of them here because you can see them in our open access publication, and it's easier if you look them up as if I just put them here on the screen. So the next topic is toxicological testing. And many of them are standardized tests, but then they're still using non-standardized materials in them, like FBS. And you can see that there's definitely a problem there. So therefore, they have to be standardized too with standardized materials. And there are studies out there that show that already for many OECD test guidelines, the FBS and other animal-derived materials can be replaced, like the ones on screen, like estrogenic activity, the test for genotoxicity, and the test for fish acute toxicity. And also for the in vitro cytotoxicity tests that is necessary for biological evaluation of medical devices, an FBS-free medium is published. These tests are mostly used with L929 and CAAT2 cells. And there's also this year published a strategy to test the toxicity of nanomaterials, also used with HPL, and the ACUTO-X method that predicts the acute oral toxicity is used with pooled human serum. So sooner or later, when you work with cells, all of us have to froze them at one point. And many times the FBS concentration in this freezing medium is up to 95%. The reason is because FBS has a high protein concentration. This helps in protecting the cells, plus a little cryoprotectant like DMSO or glycol is added. But you see here, 95% is really high. Normally, standard cell culture is being grown in 5 to 10% of FBS. However, the big thing is actually really the high protein concentration. Therefore, you can use a xeno-free medium and just put in a high protein concentration and use it as a cryopreservant instead. For example, PBS with 20 milligram per milliliter of human serum albumin and an additional 5% DMSO work well. You can, of course, try it out if you maybe put 7% DMSO or a little bit more, a little bit less of human serum albumin, but this should do the trick. But if you do not want to use proteins, you can also use polysaccharides, like in this, this research where they used freezing medium for testis interstitial cells. They used the F12 medium, put in dextran 40 and DMSO. So now you know how to replace FBS with FBS-free medium, but how to do it best, because you have your cells that are grown, that are living in this FBS medium. Now you want to switch them. How do you do this best? Well, there are 4 strategies. One of them is you gradually decrease the concentration of the FBS medium until you end up with 0% FBS. Other way, you could do a sequential adaptation while you're slowly replacing the FBS over several successive passages. You can also use the medium from previous passages to ease the transition to the FBS-free conditions in your medium. And of course, you can also just do it rapidly. You can just put them immediately into FBS-free medium. However, if there's no experience of how your cells react, you just have to find it out yourself, unfortunately. But once you transition them and everything looks good, you should maintain them at least in culture for 3 to 5 passages to ensure a stable growth and a high viability of the cells. So there are also many challenges to the replacement of FBS. Like let's say you want to replace FBS, you look what is out there on the market. You see one certain medium that serves you, you buy it, but maybe there's something in it that you don't know or you do not want, like a hidden animal-derived material. And most of the time, this hidden material is bovine serum albumin. It is frequently used in many cell culture medias, and it is not well communicated in many times if it is part of it, like in the supplement B27. There's, of course, also xeno-free B27 out on the market, but if you only see B27, you maybe don't even know what's in it. And there are also other places where the BSA is even hidden after even more layers of trade names, like in knockout serum replacement. It looks good. It seems like, yeah, it replaces serum, but one of its components is actually albumax, which itself is lipid-rich BSA. And this is a little bit ironic if you use serum replacement and then use something that is actually very high concentrated in actual serum, like albumin. So that is the big problem if you have commercial medium that is not disclosed, because you don't know what's in it. And then it remains largely unknown if nothing is reported. So if you have something where you have BSA in it and you want to replace it, and you can replace it, I would recommend you use recombinant human serum albumin. So there are also other replacements out there. If you're looking on Google Scholar for animal-derived replacements, you also find many studies that use other animal-derived materials as a replacement for FBS. For example, you find studies on using egg whites, milk whey proteins, the ocular fluid of bovine eyes, even the fluid of earthworms or other invertebrates. Others also say instead of bovine serum, you use non-bovine serum like from goats. Some even says you can hydrolyze meat tissue and use it instead, or instead of human platelet lysate, why not using bovine or porcine-derived platelet lysates instead? Well, some are getting even more creative by supplementing FBS with even more animal-derived materials. For example, I found a paper where they were using extracts from the ovaries and even the eye stacks of shrimps. So, as you can think, all of this will just reintroduce all the disadvantages of animal-derived materials back into your cell culture. And it will only shift the ethical issues from bovine fetuses to other animals. So in the end, you have nothing wrong. You just have a fancy new material that you can use. So also algae and plant-derived materials are also on the market. And mostly it's made from red algae, soy hydrolysates, or even agro-industrial waste from plants. And this is a good thing actually, because they contain a lot of amino acids, vitamins, and essential lipids that are necessary for cell culture. Because also if you are an animal or a human, you can eat plants also to make your cells grow. However, there's still a lack of standardization, especially when you use the agro-industrial waste. And they're also potentially containing anti-nutrients that could be problematic for cell culture. Therefore, it is less suitable for scientific research, at least so far in the moment. Maybe in the future it will be better. But for cultured meat production, it is an option for sustainable manufacturing. However, due to the complexity of the hydrolysates, we need to do proteomics and peptidomics, because we need to have a quality assessment and also any information on potential allergens. Because you do not want somebody who is allergic against soy to have any allergic reactions if you would eat your clean meat. So to sum up these challenges, I can say that xeno-free is key. Because when you're using chemically defined medium, but you need to have proteins. You should always use recombinant proteins at best, because this will also reduce the variability and minimize the contamination risks. And because if you would use xenogenic materials, meaning materials that are not from the species of what you're researching, then you actually are creating a chimeric environment. And I also read a paper that were actually going a little bit to the extremes with this. They were trying to, or they were actually not trying, they were culturing simian cells with bovine serum in a murine matrix, so from mice, and they detached the cells with a crustacean-derived dissociation agent. And in the end, they wanted to use this research to make conclusions to the situation in the human. And of course, all of this will create experimental results with a high variability and a very questionable transferability toward the biology of humans. Therefore, xeno-free is key. So we have to change the paradigm, and not only the researchers, but also reviewers and editors must do their part. Like the scientists should proactively state when they're using animal-derived materials in their study, and they should also write the ethical implications of that. And they need to justify it and also say that they are planning to replace them. And journals and reviewers should also demand the use of animal-free materials for in vitro publications, and also a clear scientific justifications when the animal-derived materials are still used. The journal Altex is already advising to replace animal-derived materials, and also it requires authors to justify a continuous use and potential alternatives to discuss this in the discussion section. So more journals should take example from this. However, trust is good, but transparency is always better. And manufacturers and regulators can also do their part, and they should label the cell culture products as free from animal-derived materials and make it clear and obvious for everyone to know that what they're buying. And additionally, they should also introduce a variability score to see how much variable the composition is of a material. So this can also significantly contribute to the reproducibility of science. And also cell line providers should remove animal-derived media from their recommended cell culture conditions on their websites and on their product manuals. So, as I said in the beginning, a xeno-free cell culture is in fact possible. Just like it is possible in the future to replace animal experiments. And just like with the animal experiments, a regulation is needed. Best would be if, as soon as it's possible to use an animal-free material, the corresponding animal-derived material must no longer be used in scientific procedures. Just as it is the law in the European Union that if there's an animal-free method available, you are not allowed to continue with the animal experiment, at least on paper. And in completion, also we need projects and funding for the development and the validation of animal-free materials. So, in conclusion, we need a complete animal-free cell culture science, because replacing procedures on live animals is essential, and also keeping NAMs animal component-free. Just like replacing live animals, a truly animal-free cell culture is, in fact, the goal. Because this will save money and time in your lab, increase the reproducibility and safety of your research, and enable a truly ethical science. I'd like to thank everyone who was helping to make this research possible, especially Stina Oretsen, who did like 99% of the lab work and The medium is also named after her, Athena Marek Pamlet, and many others whose names would not fit in here. And of course, the funders who gave the funding for it, like the Swedish Breast Cancer Research Foundations and the Renate Bentlin Stiftung für Nutztierschutz. Thank you very much. Thank you very much for your attention. And here you can also read the full paper.